the critical bottlenecks is they're essentially all over the process. Really starting with right at the beginning is the DNA, whether customers are using random integration. That can be a bottleneck because ultimately, if you're integrating your DNA randomly, then you don't know where it's going to go. You're going to have essentially this needle in a haystack aspect. You're trying to find that good clone. So essentially, that's where it all starts, is right at the beginning is your DNA and your choice of methodology, and random integration is perfectly fine. And that's the way the market started out, is using that.
Hey there, biotech innovators. Welcome to the Smart Biotech Scientist Podcast. I'm your host, David Bruhlman. Today, we're exploring the fascinating world of cell line development with Andrea Gough, who is the Senior Director for the Solentim Portfolio Advanced Instruments. With 15 years in biologics development and a passion for enhancing cell line development efficiencies, Andrea brings a wealth of knowledge about the evolution and future of cell line development. From bottlenecks to breakthroughs, we'll explore how modern approaches are revolutionizing the field. So grab your coffee and let's get started. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist Podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Bruhlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Andrea. Thank you for joining us today.
Thank you. Thanks for having me.
It's a pleasure having you on the Smart Biotech Scientist Podcast. Andrea, share something that you believe about bioprocess development that most people disagree with.
Good question. I think it's more the other way around, or actually you could phrase the question in both ways. But yeah, this is a really good question. Hopefully I don't offend anyone. But essentially, I would say in my career, there's been this reoccurring theme whereby CLD teams have always been trying to draw correlations between early sort of analytics, like really early stage analytics, and late stage, like bioreactor analytics. So in cell line development, the ultimate aim is to get to that cell line as fast as you possibly can. And so I always see this theme occurring with probably more startup companies and newer companies to CLD, or people that are bringing it in-house and not using, say, a CMO, where they want to try and use data to determine if there's a connection or a correlation between like literally a handful of cells and how it's going to perform in a bioreactor. I may well be out of touch, but I just don't think you can draw conclusions or correlations at that early stage. Literally, like I said, a handful of cells, even one cell, and how it's going to perform in a bioreactor. I think once upon a time, there was some data that maybe an expert in the field from Lonza published where they were saying there is no correlation. And I just think from my early days, that's always been the question. I don't think there's any correlation. And I don't think people should waste their time trying to find it, because ultimately what it means is you have to screen loads more clones and take more clones through the whole entire process to see if the ones that did or didn't perform actually correlate. So I think that maybe that's an opinion that won't go down well, but I always disagree when I see people trying to do that in the field. I think, oh, you might be wasting your time, but who knows? Maybe there's data I haven't seen out there that shows there's a correlation.
Yeah, you're making a good point, and we're going to get to the nitty-gritty of cell line development just in a minute. For now, Andrea, can you paint us a picture of your professional journey that brought you to doing the work that you're doing today? And what were some interesting pit stops along the way?
Yeah, good question as well. So my career started out in microbiology, actually. So I was very interested in genetics in my degree and bacterial genetics. And I thought I'd go down to medical micro, that route. But after university, I essentially knew that I really didn't want to do any further education. I was done with further education and I just wanted to get stuck into the science. So I started out in a small startup developing a screen for hospitals for MRSA, so methicillin-resistant Staph aureus. That was basically a company providing a solution to hospitals, and I absolutely loved that. But after several years, I realized that I really liked the training side. I liked the application side of the science, and I ended up moving into, again, another company that was in the CLD arena. And they developed instrumentation for cell line development and helping people screen clones. So straight away, I got stuck into being an application scientist and really learning all the instrumentation that company offered to support the CLD process. So that's where it all started with CLD, was way back in, I think it was like 2008 or 9. And I learned everything on the job. I was constantly with customers and learning everything about their cell line development and their challenges and almost troubleshooting with them quite a lot. And so did that for several years on a regular basis, traveled the world, got to see loads of customers and really work with them on helping them with their CLD challenges. I'd say the only pit stop that I've really had along the way is that after so many years in applications, I really wanted to make an impact on the products, the way in which we could help customers and develop the products more. So I wanted to get into product management. And with that, an opportunity arose at Thermo Fisher. So I ended up working for them as an associate product manager and antimicrobial resistance space. So almost going back to what I started out in. So I was product manager for a short period of time, massive learning curve on antimicrobial resistance in the clinical setting, and also product management. That was my first taste of product management, but I could have a real impact on the product, how people use it, and how it helps people in getting the data they need. But with that, I ended up following that product management job, I moved on to essentially the company I'm back at now is a small company called Solentim, and they developed cell line development instrumentation. So I was a product manager there. Small company at the time. They developed instrumentation, like I said, for imaging, dispensing, product quality attributes now, and also we have reagents. But I really started out when that company was small. Again, same kind of role. It was really helping CLD customers with their setup and implementing the process and just helping them with like some of the pitfalls that we often experienced of CLD in general. I also took a slight detour to another company, Horizon Discovery, where I was the product manager for the CHO cell line that they licensed. So all in the same field. And again, working with customers on the challenges that they often had. And it always sort of gravitated towards cell line development and support of the early process of cell line development. So that's why I am back now at Advanced Instruments. I am managing the Solentim Portfolio again, so I have a team of product managers and just really getting stuck in with a lot of the same customers that are still doing CLD, but also a lot of new customers and CDMOs. So yeah, that's the potted history.
Fantastic, Andrea. Let's start with the basics of cell line development. And probably some of our listeners are not familiar with all the nuts and bolts of cell line development. So can you explain what is the criticality to find an optimal cell line and ensure clonality? And what are some critical bottlenecks or difficulties when developing cell lines?
Yeah, that's a really good question. I mentioned bottlenecks earlier as well. So I'll try and approach it in two stages. So the bottlenecks, the critical bottlenecks is they're essentially all over the process. Really starting with right at the beginning is the DNA, whether customers are using random integration. That can be a bottleneck because ultimately, if you're integrating your DNA randomly, then you don't know where it's going to go. You're going to have essentially this needle in a haystack aspect. You're trying to find that good clone. So essentially, that's where it all starts, is right at the beginning is your DNA and your choice of methodology, and random integration is perfectly fine. And that's the way the market started out, is using that. But essentially what that means, like I said, is you've got a whole massive population of clones where the DNA may or may not have integrated appropriately. It could produce you high producers and so on, but you have to screen all of those. And what that means is you've got a high number. That's a bottleneck. Transfection recovery, so after you've done your transfection, can be a problem. Not for everyone by any means. It all depends on the cell line you're using, your processes, and also the molecule that you're trying to express. And have had some customers that have said it can take 4 weeks to recover that population after transfection. So that can be a bottleneck that people have to work through and try and drive efficiencies around that. The next one, after you've got your pool and you want to find that needle in a haystack clone that's going to be your great producer, you really need to determine that it's clonal. And this is really for the regulators, it's a lengthy process. Once upon a time, people used just statistical analysis to do this. But now with the technologies out there like imaging devices and dispensing devices, it enables you to dispense efficiently and then also confirm that you've got your single cell to start with. So there's definitely challenges and bottlenecks around there. And then clone screening and selection. So this is actually a massive part of the process. You've got multiple clones that you are continually looking at. The growth, are they clonal? Are they producing a lot of protein? Then you transfer them on, and then you look at other things like the product quality, and later on down the expansion line, you want to assess what they're like in a fed batch situation, because that replicates the bioreactor. There's loads of bottlenecks in terms of making all of those steps as efficient as they possibly can be. The other one that I haven't mentioned yet is clone stability. Once you've selected a whole range of clones, will they be stable enough to continue to produce that protein over a longer period of time in the bioreactor? And so this is why it's quite an involved process, and people really have to, if they're using random integration, they have to select lots of clones, screen lots of clones, grow up a good number of clones, usually in the region of 10 to 20, and then do the stability studies at the end so that they can get the handful of clones, like 3 to 5, that will then be good producers in a bioreactor. That sort of answers your first part. But the two, like the critical parts, or the criticality of optimal cell line, of getting an optimal cell line and clonality, is kind of twofold. The first part, the optimal cell line, is really, like we said, it's getting that production, finding the best producer, the most robust in a bioreactor sort of manufacturing situation, but then also the most stable, and making sure it's producing good products. Ultimately, the product quality is really important. So that's sort of the critical aspect of finding the optimal cell line. But then ensuring clonality is really important for the regulators. It essentially ensures that there's less risk in that clone being unstable. I say clone, cell line now, being unstable in the manufacturing setting, so that if they say approve that drug, you've managed to get it all the way through clinical trials and they approve that drug, the longevity of that drug in the market, they want to know that they've got the best chances possible. That's my sort of layman's term look on it.
I'd like to dive into the clonality, because that's obviously a very critical aspect as we are working in a highly regulated environment. So we have different methods or tools at our disposal. And some of you listening are probably familiar with the limiting dilution. Now we have a lot more modern tools. Can you help our listeners understand what different tools we have at our disposal, and also what different approaches you have seen now in your long career already, and what approach has really impressed you?
Yep. So limiting dilution has definitely been the staple. Anyone could do it. Essentially, it's plating cells at a really low density so that the statistics are that you will get a single cell in a well. But the densities are so low that you have to essentially plate loads and loads of plates to ideally get one cell in a well. The challenge with limiting dilution is often cells stick together and there's no means of separating those cells so that you get it into a well on its own. But like you say, technology's come quite a long way. So when I first started out, I mentioned I worked with a company that was developing instrumentation. They did the semi-solid media method. So that's where you take a whole load of cells and then you plate them in semi-solid media. And that semi-solid media keeps the cells separate as they form into a clone. So this was a really good principle. And then that device would essentially pick that clone and then take it on to the next step. But the challenge that arose was this clonality, where people couldn't really see, the optics weren't high enough to see that clone started from a single cell. You could only really see the clone. So more proof was required. And then so after that, you would still have to do limiting dilution.
That was the clone picks, right?
Yes, that's the clone picks, exactly. So they had clone picks and they had the FL aspect of it. So as the clone was growing into a clone and producing protein, there's a capture antibody that would capture the antibody. And then you could see a fluorescent halo around that clone. And then essentially you could pick that clone knowing that it was secreting the protein of interest. So great technology, but did unfortunately have some of these limitations that you would need this extra round of either limiting dilution or some other method, single cell dispensing. FACS was also used. I kind of feel like FACS is like a hammer to crack a walnut. It's great and it definitely sorts the cells, but it's quite harsh on cells. Some cells are robust enough to cope with it. But one thing I found was that it's quite complex technology. You need an expert and it was harsh on cells. So people, whilst they could use it, it wasn't ideal. So there were pitfalls with FACS. So because of those challenges, and then I mentioned earlier that imaging devices came about and they helped the process with proving clonality. The next bottleneck was single cell dispensing that actually produced a single cell. So there's loads of seeding devices out there in terms of some really cheap and cheerful ones. I'm not convinced all the instruments out there will suit most people's needs. But yeah, there's single cell devices out there that have improved the dispensing efficiency. So that's the first bottleneck, is actually getting more cells in more of the wells so you have fewer plates to deal with. So once upon a time, people were screening 50, 100, 200 plates of limiting dilution. Now that's got down to less than 10 because the seeders are more efficient. Where I work, we have our own technology. I'm biased. I think our technology is great. It provides images of the seeding dispensed in the well. Then it fills up the actual plate for you, so you don't even have to do that. And then it also captures the whole well image. So yeah, have I answered every single question in that? I think you were saying what challenges were solved. Hopefully I've answered that throughout.
And what I'm hearing, Andrea, is also, obviously there are lots of different technologies out there, like a FACS that works really well, but is complicated. There are easier ones now. There are a lot of new imaging tools or simple dispensing tools. Can you help us understand? Because I think there is no one size fits all approach, right? It really depends on the application or how many cells you're going to screen, or whether you're a small startup or you have a huge infrastructure. What are some common misconceptions around choosing the right equipment?
I think misconceptions, I think that flexibility is key. So you really want a device that fits your needs and gives you all that documentation and evidence that you need. I think that's the key. All the devices have their benefits. The majority give you good seeding efficiency. We certainly do. In my mind, it's making sure that the instrument meets your criteria, your flexibility needs. You don't want to be stuck doing their methodology. You just want a device that does what you need it to do as fast as it possibly can. And also, I think tracking the clones is really important. And also, as much evidence as you can gather is also really important. Totally, for the regulators, the more you can provide in terms of real evidence that you started off with a single cell and then formed into a clone, and that image is clear as it possibly can be, will really help avoid questions, I think.
Cell line development is in general quite a lengthy process. We're talking about several weeks or if not several months. Let's talk about the timeline pressure, because a lot of people, like when you're at conference, you see, well, we reduce development timelines, or like we have X amount of months to first in man, or these development approaches. So what innovative approaches have you seen talking to your customers they use to accelerate development while obviously maintaining quality?
Yeah, so I would say there's been several over the years. I think that the main thing that I'm seeing is people trying to take things off of the critical path. So in the CLD, like I mentioned earlier, there's lots of steps in the process. But if you can take things off the critical path, like stability, then that will help you. If you can screen fewer clones, that will help you. So I'd say some of the things that really help with time pressures is things like the transposon technology. So, semi-targeted integration will definitely help people screen fewer clones. And also, because it is semi-targeted, the stability is not given, but it's definitely, there's data out there that shows that the stability is a lot better, because you're essentially putting your gene somewhere that is not just going to be randomly lost or cut out for some reason. It's, like I say, it's semi-targeted. So I think that's definitely improved things for customers. Not everyone can use transposon technology for whatever reason, but it's definitely improving people's processes and timelines. With that, the other thing, like I said, with the ability to produce clones using transposon technology, the semi-targeted, you could potentially take the stability aspect offline. I think you'd have to do a project to prove that the stability is good so that your process is validated, so to speak. But yeah, the other thing that I've seen is using bulk pools as well with transposase technology. And sometimes people do it with random integration as well, is you use the pool to generate some initial product that you can, say, use in preclinical studies or even initial clinical studies to try and speed up the process. So yeah, they're the things that I've seen that so far seem very innovative and have really cut back some of the timelines.
That wraps up part 1 of our conversation with Andrea Gough. Join us next time as we continue talking about cell line development and dive into practical advice for any scientists starting their cell line development journey. A huge thank you to Andrea for sharing her expertise with us today. And before we go, are you looking to accelerate your biologics development while keeping costs in check? Develop biologics better, faster, at a fraction of the cost with our fractional CTO services. If this is of interest, just book a free call with me. You'll find the link in the show note. All right, smart scientists. That's all for today on the Smart Biotech Scientist Podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.